Well, good evening, everybody, and good evening, Barcelona. It is a big pleasure for me to introduce you to Jean Campbell. Dr. Campbell received her PhD from the University of Wisconsin-Madison, and then she had a postdoc with Ed Krebs. And she has been working in many fields in molecular biology, physiology, hepatology, oncology. And in 2014, Dr. Campbell received the Nelson Fausto Distinguished Faculty Mentor Award. And I cannot agree more with that award because she's a wonderful person I had the privilege of spending 2 years and a half in Seattle. And I recommend every young doctor and every young pre-doc, if they have the opportunity here in Barcelona to go to Seattle with Jean. She will be a source of knowledge and inspiration. And then Dr. Campbell spent 3 years in a company, Oncosec Medical, dedicated to intratumoral therapies. And currently she's at the Fred Hutchinson Cancer Research Center and University of Washington, working on CAR T-cell therapy and immunotherapy. And I would like to thank the organizers, Dr. Bañares and Dr. Gracia Sancho, for the opportunity to bring Jean and also the sponsors. And without further ado, I leave you with Jean.
Excellent. Thank you, Javier. It is an honor and a pleasure to be here this afternoon, and I also want to thank the organizers. I've had a wonderful day talking to various people about their projects. They're exciting, they're diverse. I'm excited about the progress that's happening here. I specifically want to thank Javier for inviting me to come. I've had a lovely time with his family, enjoying the environs of Spain. I want to give a shout out to Barcelona. Sorry, I didn't pronounce it correctly, but howdy, Jordi. Hope you're well. So today, Javier asked me to talk about liver regeneration, and it's such a small topic that I figure we can cover in about 5 minutes. Actually, the challenge is that it's a very big topic, and I cannot cover it and do it justice. So today, I'm going to give a presentation a little bit about liver regeneration, and I am going to frame it in the Seattle perspective. There are different perspectives around this, so I'm going to talk to you about my biases so you are fully aware of those. We know a lot about liver regeneration in normal mice, which is my model organism, but in human beings as well. But I think the real next step is to understand how what's the liver regenerative capacity in models of liver disease such as fibrosis, and we worked on a model of liver fibrosis. And then we wanted to restore that liver regenerative capacity and hopefully block fibrosis, and so we did a number of genetic and pharmacological approach to block fibrosis. So I think Pablo Neruda said it best. I have an ode to the liver. I think it is an amazing organ. I love life. Do not betray me. Work on. Do not arrest my son. It is a wonderful poem. It really fills you with inspiration around the amazing capacity of the liver to regenerate. My view of liver regeneration came from the field of signal transduction. So I was working with Ed Krebs and Eddie Fisher. They were discovering the MAP kinase cascade. We were in cells and culture. And then I found out about the liver, where there was this in vivo synchronous process to understand the cell cycle. And I thought this was really exciting to actually be able to do surgery and have the synchronous process. So this is my liver perspective, is that I think signal transduction is cool, and I think that the capacity of the liver to regenerate is amazing. I need to give a big thanks to Nelson Fausto. He spent 5 decades working in liver research. Sadly, he died in 2011. Javier and I both got to work with him. For me, he was an amazing mentor, juggling the chairmanship and the responsibilities of a research lab while trying to get research funding. So I did a little digging, and this is one of the first papers I found that Nelson was a co-author on, not the senior author, and this is in JBC, and I just want to point out the date of publication versus the date of submission, and we think we have it hard to get papers published, but so did he back in the 1960s, so it's still a rough path to get your papers published. So just to set the stage, to level the vocabulary, everybody here will know, but I think it's good to say liver regeneration is really compensatory hyperplasia. It's not true regeneration. We don't regrow the anatomic features of the lobes of the liver. The liver size is specified by this optimal liver mass to body mass ratio. We don't understand those systemic endocrine, paracrine, and juxtacrine signaling that influence this back when we first started working in animal models. And to this day, we have some ideas, but they are still just ideas. I think it's remarkable that the hepatocytes throughout the lobes, not at the cut surface, they all have this ability to regenerate and to grow. So it's a uniform set of signaling. The hepatocytes are cell autonomous. Liver regeneration after surgery does not involve a stem cell compartment, and the hepatocytes do not differentiate. They maybe downregulate albumin a little bit, but they don't move into a pluripotent case. They stay as hepatocytes, providing all the enzymes that hepatocytes do. Now, as you know, there are specific cases where you need progenitor cells, and Nelson was involved in the study of that, and I heard a nice talk today about oval cells, which is what we call the stem cell population in rodents. And they emerge when the hepatocytes can't regenerate under severe conditions. And it's good that we have this. We need a liver to survive. I think it's just another redundant system that the liver has developed. Not enough time today to sort of delve into the work that Nelson or I did on oval cells, so I refer you to some review papers. So I think that the interesting thing to me is this in vitro versus in vivo conundrum. So we know from a series of experiments that spanned a couple decades that in vivo hepatocytes replicate really efficiently and with apparent unlimited capacity. Yet, if you take that hepatocyte out and you put it in culture, it loses this capacity. It maybe divides once or twice. And so you have this basic difference, and we've used both of the cells in culture and the regenerative model. But I think that we still are trying to understand how these mechanisms work, and clearly that in vivo environment holds the key. The other cells that are in the liver, the non-parenchymal cells, are really important in understanding how this regenerative capacity exists. So just to go back a little bit in history, the couple studies that I think about with this amazing capacity is from Eric Sangren and Jonathan Rim, where they took a mouse that had a uroplasmin activator transgene. It's there in panel A. It's white. It's diseased. They transplant in I think a couple thousand hepatocytes. 6 weeks later, you can see the hepatocytes are taking over, and by 8 weeks, it looks virtually no different than a wild-type liver. Overturf and Grompe did a very similar thing with a FAH model, and thank you. Thank you. So we know that you transplant a hepatocyte and it has this ability to keep replicating. It's not going to hit the Hayflick limit that we know that cells in culture meet. I don't know how they do it with their telomerase enzymes, but it has this amazing capacity. What most people in the field of liver regeneration use is a model that Higgins and Anderson developed in rats called the 2/3 partial hepatectomy model. And it's shown here where you remove sort of 70% of the liver, and over time you regenerate that liver mass. Javier tells me he's developed a better model for this, so I'm excited to read about that. But what the early studies showed is that you get these sort of two synchronous waves of these hepatocytes that proliferate, and that the non-parenchymal cells, or NPCs, follow this hepatocyte proliferation. And by 7 to 10 days in animals, you restore the liver mass. In humans, it takes a little bit longer. And of course, this is when there's no disease in the liver. But I thought it would be good to just, again, go back in history and look at some of these earlier techniques. In this experiment, Grisham injected mice with tritiated thymidine and then performed a partial hepatectomy to ask about the label of cells that says % the cells labeled. And you're looking at the labeling with tritiated thymidine, very labor intensive. But you can see that a couple of features that are critical is that there's no incorporation of thymidine to the DNA synthesis, and then you get a wave of hepatocytes or parenchymal cells, followed by the hepatic stellate cells or liver endothelial cells or Kupffer cells. So they follow. You can also look at the cell cycle by looking at hepatocyte mitoses. They're very easy to spot in a liver H&E section. It's much harder to look at the mitotic indices of the non-parenchymal cells. But this basic approach was used for decades to try and understand what was going on with liver regeneration. So this is where I'm going to sort of insert Nelson and the ideas that he had, is that in the 60s, we were still trying to understand what the cell, we're all trying to understand how the cell cycle worked. And of course, there were basic biologists who were working with yeast to understand the division, and they had mutants. And so we had the orthogonal approach from yeast and cells and culture. And this fit in really well with what was going on in the world of liver regeneration. So the hepatocytes in a normal liver are quiescent. They sit there, they wait until there's a stimulus to progress into the cell cycle. And so Nelson and colleagues did a number of experiments to look at the priming of this, because he hypothesized there were some factors that caused this cell, this hepatocyte, to move into the G1 phase. And we know from these experiments there's a restriction point. You have enough energy to synthesize DNA, to synthesize your ribosomes, your proteins. And so this is a checkpoint that was identified in yeast to say this is an important point. If you go past this point, you're committed to the rest of the cell cycle. So Nelson hypothesized that there were cytokines that were involved in this, and did a series of experiments to show that, yes, indeed, you can push hepatocytes into this phase of the cell cycle. And this was infusing cytokines and growth factors. But then we began to define the different phases and the factors and pathways important to the progression, as well as the termination, because after you reach this optimal body size, you cease, you stop dividing. Some hepatocytes divide twice, some hepatocytes divide once. It's not the same hepatocyte that goes through 2 rounds. It could be another hepatocyte that picks it up. And Eric Sengren did this fascinating experiment where he transplanted mouse hepatocytes into a rat and did partial hepatectomy. And rats and mice have a slightly different timing, and they maintain their same timing of regeneration. So again, an interesting transplant model to help us understand about the cell autonomous nature of hepatocytes. So really the next component that was really important to sort of understanding these phases of the cell cycle were the use of transgenic and genetically modified mice, systemic knockouts, cell-specific knockouts. And this is where I'm going to embarrass Javier, because he was rather busy in the lab in Seattle, and we had done with a number of people, and I'm going to apologize right now to the many people who did work on knockouts, cell-specific and systemic knockouts in areas that I don't have time to sort of go over. But this is these animals are from Javier's 2011 hepatology paper, and you may not recognize him, but this is what he looked like when he started in Seattle. He always wore scrubs because he was constantly doing surgery on this many different animals. Again, it was a pleasure to have Javier in the lab. A nice book that was written by some of us in the field. This just highlights how many people have been using these genetically modified mouse models to understand the different phases of the cell cycle. And so we heard about cyclin E knockouts today. We've heard about SOS, son of sevenless animals. People continue to use these knockout, either cell-specific or systemic, to try and understand this basic process, the initiation, progression, and termination. And I think what the take-home that I would say from this point is that I have not seen a paper where you have knocked out a gene that has demonstrated that it's necessary and sufficient. So this is a genetic term that you have to have necessary and sufficient. We have impaired, we have modulated, we have blunt, but we have not found a knockout where you've grown up as a healthy mouse, either induced the knockout and then had the animal die because they couldn't regenerate their livers. If anybody knows of some, I would love to hear maybe a question from Barcelona at the end of the talk. But I think it's a very interesting thing is that the liver has figured out a way to regenerate, hence my ode to the liver. If we all could figure out this, then this would be amazing. So through the use of genetically modified mice, we kind of stepped away from looking at hepatocytes. We were fairly hepatocyte focused, and then it became really important to look at the non-parenchymal cells. And there's a lot of interesting work in the last decade looking at the role of non-parenchymal cells and the timing and cessation of liver regeneration. And again, this is a model that we use a lot to sort of understand what's going on and the factors that slow it down. We've knocked out glutathione reductase, and what we do is we slow the regeneration until they get enough resources to get ready to build their building blocks to go into S phase. So everything sort of makes sense with this sort of framework of the cell cycle. Okay. And again, what got me interested after coming from a signal transduction lab where we were defining what the MAP kinase cascade was this ability to do a partial hepatectomy. This is in mice. We have sham, which is just doing a laparotomy and closing it back up, and looking at the waves of phospho-ERK, cyclin D, PCNA, and of course, because I was interested in all the MAP kinases, I looked at the phosphorylation of P38 MAP kinase, and I thought it was intriguing that this enzyme actually got dephosphorylated right after liver regeneration. So these waves led Kim and Nelson and I to sort of come up with this cartoon. These are all the different factors, pathways that need to be coordinated so you can move through the different phases of the cell cycle. And again, I can't list all the different knockouts that were used to help define these sort of waves of cytokine, growth factors, and the metabolic input. But of course, we now know that the microbiota, what's happening in the gut matters. Again, a very nice presentation to me today. The metabolic conditions, what happens with obesity, how well does the liver regenerate in this? And I do want to point out that when we use genetic models, we do have some bias there, right? So you have some adaptation because of the genetic modification. And so some people in the field, including George Michalopoulos, have been using the shRNA approach to do an acute deletion of a gene or a growth factor and ask questions about regeneration in that. So again, orthogonal approaches to address how you understand a physiological phenomenon. All right, there was a bit short shrift to liver regeneration. Sorry, I'm the speaker, so I'm going to talk a little bit about the research that I do. So in this case, we've used a reporter mouse that is expressing a nuclear GFP under the expression of platelet-derived growth factor receptors. And so we're looking at the stellate cells in this fresh section that we just stained, just cut, fixed, and put in agarose and did some imaging just to sort of look at the beautiful liver. So I'm going to tell you a little about the platelet-derived growth factor C mouse that have been studying. So of course, we know a lot about liver regeneration in the context of a healthy, normal liver, but most of the time what we see is injured livers, inflamed livers, diseased liver, and there are a variety of ways you can get this liver injury, and of course it's a insult that can lead to fibrosis. We can go back and forth along this. I believe it's reversible if we don't go too far. The double-headed arrow leading to cirrhosis is unknown, but I still hold out hope that we can reverse it. And of course, cirrhosis is a primary stage to set up for hepatocellular carcinoma. And so in the context of this talk, the question is, what's the regenerative capacity in here? What's enough? How can we restore it? Is there a continuum along this progression pathway? It's sort of a big question that we want to address. So in the model that I use, it's a genetic model. We overexpress platelet-derived growth factor C. So there are 4 isoforms of this ligand, and this cartoon just shows the receptors in the transmembrane space. The little balls represent the different forms of this growth factor. So PDGFC is excreted from the cell as a dimer, a heterodimer, but it's got this cub domain represented in orange that has to be cleaved when it binds to the receptor site. And we think the tissue plasminogen activator is one of these. But there's also another isoform called platelet-derived D. And so in this model that I'm going to tell you about, when we overexpress this under the albumin promoter, we get this fibrosis that progresses to angiogenesis and cirrhosis and hepatocellular carcinoma, as a mouse could have, over the age of the mouse. So some features of this model are some immunohistochemistry. So we've got wild-type mice here looking at alpha-smooth muscle actin and glial fibrillary acidic protein. Here's our transgenic mouse with the sort of sinusoidal expression of alpha-smooth muscle actin, 7 weeks, 4 weeks, and then here we have glial fibrillary acidic protein. I think what's interesting is you've got the different kinetics of these 2 markers of fibrosis, of activation of the hepatic stellate cells. So we felt that the platelet-derived growth factor wasn't binding to hepatocytes. In our hands, they don't express the receptors, but the stellate cells and the myofibroblasts do. But I think what was nice about this model is that by 9 months, you see from a trichrome staining this kind of chicken wire fibrosis that's there. It's very profound here, and then you get this multifocal presentation of the cancer, and the liver enzymes are elevated. This time point is 9 months. So we've got an event of a transgene is being expressed that's causing activation of stellate cells, inflammation, which I'm showing you only a part of here. And as you might expect, with that much liver burden, that these animals succumb to their liver disease earlier than wild-type animals. So it's a nice model because we have this early phase that we can look at. Hopefully we can intervene to prevent progression to the next phase. So this cartoon just sort of summarizes what we know about this model. We've got sort of our normal hepatocytes. We get this expression of this growth factor. We are seeing activation of stellate cells and fibrosis. We see dysplastic nodules, and then eventually by 12 months, hepatocellular carcinoma. And we see an increase in collagen and extracellular matrix. The cellularity increases. The hepatic stellate cells clearly are fibroblasts. We have features of activated endothelial cells. We've done a lot of kinetic studies at time points with gene expression and immunohistochemistry to see a lot of factors that go up. But the real importance of having a model, it's a model, is to ask some questions about what happens. In this case, we wanted to ask about what happens with liver regeneration. So Javier and Kim did surgeries on these mice at 8 weeks in this thing. And what you're in this figure, in A, what you're looking is at the BRDU, a pulse of this label of the DNA, is an ability to look at the time course of incorporation and the proliferation of hepatocytes. And what you can appreciate it compared to the white open bars, which are wild-type mice. In the transgenic mice, you see a real suppression of the liver regeneration. And you would think that these mice were goners because they really haven't done anything to restore their liver mass. But in fact, a good surgeon, they survive. They just take a long time to regenerate. So their regenerative capacity is low, but they still can sort of get by. So the mitotic figures just correlates with sort of S phase and M phase, and you can see the 2 different waves that I so much like in this model. So I told you that this model has fibrosis, and in this rather complicated slide, let me walk you through it. We wanted to ask, is this regenerative capacity correlated in any sense with the amount of fibrosis or collagen deposition that we see. And so it may be hard to see in A, but this is 5-week animals, 8-week animals, and 12-week animals. And we're looking at wild type and transgenic mice. And what we're looking at is at 5-week animals, again, Javier had his surgical hands all over these mice. We see at the 48-hour time point that at 5 weeks, there's not a significant difference. But at 8 weeks and 12 weeks, we see this profound difference in these mice. We looked at the incorporation of BRDU into non-parenchymal cells, and again, the proliferation of those non-parenchymal cells is most significant at the 12-week animals. We're looking at mitotic figures. And in the bottom row, we're just looking at the extent of fibrosis with Picrocereus red. So 5 weeks versus 12 weeks with a high MAG inset. And then this is just quantification. And so if you compare the black bars here and the black bars here with the extent of fibrosis with the loss of capacity, you see a correlation between this. So the more fibrosis there is, the less regenerative capacity. And this has been known and showed in other models. This is just a demonstration in this fibrotic model. So we wondered why. What were some of the reasons that could happen? And it will not surprise you that when we did a microarray analysis, that one of the top pathways that was upregulated with TGF-beta signaling. This makes sense if you've got activated stellate cells. There are a lot of other changes that go up in this case as well as down. So what we then did, or Junil did, was to undertake a heroic process of crossing the SMAD3 knockout mice that we got from Anita Roberts and generating these 4 experimental groups to ask what happens when you knock out SMAD3 systemically. And so in full disclosure, these mice aren't very healthy because they do need SMAD3. And so the project took a very long time to complete just because we had a hard time getting the genotypes. But if you look at just baseline, this is not surgery, baseline proliferation, again, you're looking at the BRDU incorporation in hepatocytes versus non-parenchymal cells, wild type versus the SMAD3 knockouts. And then you see the wild type versus the transgenic. You can see that there's that increase at the baseline of the proliferation that you see in the wild type. But that the double knockouts of, the mice that have the transgene and knocked out SMAD3 have less incorporation. So they're taking down this abnormal proliferation of the hepatocytes in the transgenic mice. And of course, the striking feature in the transgenic mice is this increase in the non-parenchymal cell proliferation. And this too was blunted in the mice because, of course, the SMAD3 was systemic, right? It wasn't cell-specific knockout. So the question was, what did we do with the fibrosis? And you can see, compared to the transgenic mice, that the transgenic knockout mice have less, not none, but less. And then we quantified it with Picrocereus red. So we were able to determine that the SMAD3 had an effect in this model. So, of course, we we isolated whole RNA from whole liver. We didn't isolate cells. And we asked where in this pathway where we have platelet-derived growth factor C pushing the fibrosis, did the TGF-beta or specifically the SMAD3 pathway intervene? And so I've divided the groups into SMAD3 dependent and SMAD3 independent. And each of the bars are going to have wild type, SMAD3 knockout, PDGF-C, and then the transgenic SMAD3 knockout in the last. And so on this side, what you can appreciate is that relative to the transgenic mice, when you have SMAD3, you have a decrease in alpha-synuclein actin, a decrease in the expression of collagen genes, and a decrease in the connective tissue growth factor. But there are other genes that were unaffected by this block in TGF-beta signaling, basically giving us a sort of insight as to where this defect was. And these would be the PDGF receptors, which are on the stellate cells, the expression of TGF-beta itself, which is causing these genes to be upregulated. That's what we'll show in the next figure. And then this marker of stellate cells. So in a summary slide, we have the overexpression of platelet-derived growth factor C, which is driving this in this genetic model. We have quiescent cells, and we've got an activated set of non-parenchymal cells. and is specifically the activated stellate cells. So when we take out SMAD3 and all of these, we see that we are still expressing the drivers of the activation of the myofibroblast, but we're blocking the extracellular matrix as in collagen. So interesting sort of dissection of the pathways that we know to be involved in liver fibrosis. Okay. So then we moved to use this model of liver fibrosis to see if we could intervene both genetically as well as pharmacologically to see if we could improve the regenerative capacity. But more importantly is, can we block these early stage phenotypes in this model that has this progression to see if we can delay hepatocellular carcinoma. So I'll take you back to the SMAD3 knockout. This was the whole purpose of the project, but because the mice were so sick, most of them died, whether just SMAD3 alone or with the transgene before we could get to understand these studies. And so really the approach that we need is a cell-specific knockout for that. Okay, so in the remaining time, what I'd like to talk about is the use of tyrosine kinase inhibitors. Again, this comes from my time in the Krebs lab and working in small and big pharma. So again, the model that we have is we're looking at this driver that's being expressed out of the albumin promoter, the PDGFC. It circulates. It causes the activation of stellate cells. I think it causes inflammation and activation of Kupffer cells and activation of the liver sinusoidal endothelial cells. So we have a loss of fenestra in this model. We have upregulation of CD31. All the cells are sort of responding to this driver. So again, just want to remind you the importance of the adjacent abnormal microenvironment, and this was really best shown by Lovett and Jordi and Hoshida way back when in 2007, and they just did an array and looked at bisected the gene expression into sort of good and bad prognosis. And what you're not seeing is the survival curve for looking at the gene expression in the hepatocellular carcinoma, but the gene expression in the adjacent tumor microenvironment, the diseased liver, but not the one that actually has the cancer. And you can divide these gene signatures, if you will, into sort of good prognosis, those that overexpressed these genes, and those that overexpressed these more inflammatory genes. It's just to highlight that the importance of the microenvironment in liver disease. So we, with Jordi, obtained some sorafenib in this study by Friedrich, and we wanted to know a number of questions. And here, what we asked is, does sorafenib treatment in these fibrotic mice, can we restore any of the regenerative capacity? It basically needs no introduction, but I want to, from a kinase standpoint, point out that it is not a selective kinase inhibitor. It can inhibit all of these kinases, of course, in a cell kinase assay in a test tube, as opposed to what's being expressed in the liver. But it's really important when we go into these models thinking that we know that we're going to hit RAF or a VEGF receptor or a platelet-derived growth factor receptor. All of these inhibitors are competitive inhibitors, and we can go back to enzyme kinetics, and you know that in the pocket of a kinase, you have to have ATP bind first, then the substrate or the other kinase binds. So if you have limiting amounts of ATP, again, a competitive inhibitor, you're going to block that. You're going to affect what you're going to be able to phosphorylate. And so the amount of ATP that's present is going to also dictate the potency of a drug. So we go into these studies thinking that we understand from an in vitro assay what we're targeting, but I would say that we go hoping that maybe in fact it's a dirty bomb, that in fact it hits a number of these receptors because they would probably help decrease the fibrosis or angiogenesis. So I went into these studies with an open mind. I wasn't thinking we'd hit RAF. I didn't think we'd hit the VEGF receptor because I didn't know the ATP concentration. So we dosed the animals, and this is no regeneration in this case. This is just the PDGFC that overexpress the platelet-derived growth factor C, and we have the elevation in the labeling of the hepatocytes. So we dose the animals once a day for 7 days. We wait 12 hours, and then we pulse with BRDU, and we're looking again at incorporation into hepatocytes. And in this case, in the pairs, we're looking in the black bars is the addition of sorafenib in the livers. And so what you can appreciate is that there is an inhibition of the hepatocytes to incorporate BRDU or proliferate, but the more striking effect was on the non-parenchymal cells. And in this assay, it's just all the non-hepatocytes. There was no deeper interrogation of this. We have a difference. What is this difference? Because we were surprised. We then wanted to make sure this wasn't something that was unusual about the fibrotic mice, and so we did the same treatment as many others have done in mouse models. Again, we're not. This is after they're treated with the drug. They take a drug holiday for about 12 hours. It doesn't clear the systemic levels, but what you can appreciate at these multiple time points is that in wild-type mice, no fibrosis. That there's no difference, really no statistical difference in what happens to the hepatocytes. And you get these interesting sort of waves that happen with the drug. Is sort of what looks like an increase, but they weren't statistical. But then we dug in more specifically after partial hepatectomy, and you now see that we're looking more deeply at this time point after partial hepatectomy. So I think I must have made a mistake in the previous slide. But what I want to point out is that you've got this wave of proliferation of non-parenchymal cells, and that sorafenib really blunts that proliferation. So the question was, what's the non-parenchymal cell that sorafenib is inhibiting? So we do gene expression. In this case, we turn to the NanoString platform. They have defined platforms, and what I show you on the left is a volcano plot. And so basically, we're comparing PDGFC mice and PDGFC mice with sorafenib, no surgery. And what you can appreciate is around the zero, here's the sort of volcano, is there's a ton of genes that are downregulated, and they're significantly downregulated. But I was most interested in trying to identify the cell types that were being targeted by this. And so we used this approach of NanoString, which is very robust in the FFPE sections, because we can talk about the technology, but it works if you can extract RNA from FFPE blocks, and it's a great tool. But what we are looking at is sort of markers of the cell. So these are normalized cell counts. We normalize to 15 housekeeping genes and ask what's the change. And again, we have wild type with sorafenib, transgenic mice, and then transgenic mice with the sorafenib. And what you can appreciate is that F480, you can see that with sorafenib in the wild type mice, you see this big decrease, and this is also seen with the change from the transgenic and down. And if you look at CD11B or CD11C, you can see the upregulation between wild type and transgenic, and that you see a decrease with the sorafenib. So we're hitting a lot of myeloid cells, right? We're affecting them. So want to complement this approach, the gene expression, with another approach. And so we used reporter mice, and we haven't false colored this image, and so we just have an N of 2 here. In this case, we're just looking at the wild type mice, and we have a reporter gene for the PDGF receptor, and so the little dots are the image, and you can see that there's really no difference with a stellate cell, but what you can appreciate is there's many fewer cells for F480 that are expressing F480, and at this point we don't know whether we've just lost the epitope or we've lost the cell. We can't tell from these studies of this. It just highlights that. So we then did an intensive sort of immunohistochemistry, reporter mice, and multiplexed immunohistochemistry to sort of go through make sure that a single marker does not a cell make. And so we were looking at the F480, which I just showed you. We stained with MAC2 or galectin-3. We had Tie2 reporter mice, etc. You can read the table itself. And really what stands out is that with sorafenib, whether it was a wild type mice or a transgenic mice, we saw a decrease in these markers. And so we did, from a gene expression side, want to look at what was going on with collagen. And again, you're now used to seeing the wild type and the transgenic increase, the effect of sorafenib in the wild type or sorafenib in the transgenic. And you can appreciate that the collagen gene expression is going down. And what you see here is that the alpha-synuclein actually appears to go up, at least at the gene level, and the receptors for the PDGFCs are not significantly changed. So a data I'm not showing you is that we saw some histological decrease, but there was a lot of scatter in the data. So again, we can modulate the genes, the gene expression, but the fact that there's collagen deposition already there, we're not cleaning up or metabolizing it, is going to be the challenge, right? Is we have to get rid of the collagens already in the extracellular space. All right, so a cartoon to sort of diagram what we are seeing, and so we went in with an open mind about where sorafenib was going to be targeting, and this is in the fibrotic mice. This is my framework, and so what we have is an activation of a number of different cells, and we have a number of populations that are expressing F480. What I think is maybe happening is that because the CSF1 receptor is in that same kinase family. Go back to your kinome. It's in the same family. It wasn't on the list of kinases that were tested, but I think that what may be happening is a similar phenotype that you see in the OP mice, is that these mice lack this receptor, and we may not be getting we may be getting a specific inhibition. I don't know if it's at the bone marrow or the self-restoration of the tissues, the Kupffer cells. This is where we want to go next. Before I go to this sort of summary slide, I think that I want to stop here and think about it, is that a lot of people in the liver field use flow cytometry to sort of analyze what's going on in the cells. Here we treat with sorafenib and we lose the epitope that we'd want to use to help pull it out. So we were left with a technological challenge. We tried our, again, and also doing perfusion in fibrotic livers. This is a challenge. Your yield is poor. You don't know if you get recovery of all the different cell types. So we thought a long time about how do we tackle this problem. And so I'm going to come back to that. But I want to leave you with is that the field really has learned a lot from flow cytometry. But you always need an orthogonal approach. All right, so we've used these mice to understand a lot. We've used a number of kinase inhibitors as well as sildenafil. We'll talk about the little blue pill and how that works. But we've also done, we saw a dysregulation of P21, and so we crossed the mice with P21 and hope to publish this soon. But we're looking at the effect of fibrosis and regeneration and what we think is going on with the different liver cells. And so I had previously published that we saw a change in the CD31 after treatment with imatinib. With sildenafil, what we saw was a decrease in alpha-synuclein. In sorafenib, we have this phenotype where we lose the F480. So what I think, again, another pause is that if we're targeting kinases that are actually on hepatocytes, c-Met, EGFR, we are not going to allow those hepatocytes, which need those growth factors, to grow. So it makes so much more sense to go after the receptors or the dysregulation of the receptors in the non-parenchymal cells, because the bystander effect, if you will take care of the injury, inflammation, cytokines that are expressed, hopefully the metabolism of the extracellular matrix, to allow those amazing hepatocytes to regenerate. Now, I don't have any data on that, but I think this is a very good approach, is to really sort of go after the non-parenchymal cells. And I think this is exactly where the field is going. So how do we address this problem that we have, that we can't pull out, that we can't isolate the cells that we want? So I turn to what is known as the ribotag mice, and this is unpublished, and it's a fun little technique, and I thought I would share it with you. So a lot of genetics, a lot of mouse colony costs, but what you have is you have these flipper sites, and you have exon 4 in a ribosomal protein. This exon expresses the HA tag, which you all know from epitope tagging proteins. So if you cross these flipper mice and you get these LOXP sites around, put the LOXP sites around the normal exon and not around the other exon that has the epitope tag, you now can use cell-specific CRE to tag these cells. So that's what we did. We bred the ribotag mouse. It's at Jackson. It was developed at the University of Washington. You tag them, and then I crossed them to the PDGFC mice. And then you can pick your CRE. to ask what you want to do. So what happens is that you've got a heterogeneous population of cells. We, of course, were focused on the liver, and that's why my flag is pointed on the green liver. And what you have is cells that are expressing the normal exon in this ribosomal protein, or one that has the tagged. So ribosomal proteins bind, ribosomes are cooking along, you have transcription translation, so you can pull out, immunoprecipitate active transcribing ribosomes. And so you get this once they're tagged. So if my hypothesis would be that if I tag my mice with lysem-cre, and they also express F480, and sorafenib gets rid of that epitope, I still have a tag. I still can pull this out. So first, does it work? Can you look at this epitope? So we used immunohistochemistry, and I've got rat Nestin-cre, which actually does express to some extent in the mouse liver. We've got lysem-cre and, of course, albumin, just as proof of concept. And so what you can appreciate is that with immunohistochemistry for HA, that I'm getting cell-specific expression at the immunohistochemistry level. So, of course, I don't have the pièce de résistance and the data, but I did want to show you this and point out that my colleague Nick Crisp at the University of Washington is doing experiments with these. Then he's using, instead of nanostringing, he's using Fluidigm. Okay, so he helped me do this. So these are the panel of genes that he has in his Fluidigm panel, and you have your reference genes, your genes involved in repair. And again, these are all air quotes, right? Because we're talking about a very plastic cell population. So we have antigen-presenting cells, your quote-unquote M1, M2 type, but you can look at the expression, the enrichment factor, so you get total RNA, and you look at that, what's enriched in your immunoprecipitated HA-tagged cell. In this case, it's lysem-cre. So let's contrast. You've got flow, where you perfuse, you do centrifugation, you do flow, you do sort, how many hours have gone by, or you sack the mice and freeze the tissue and homogenize. So the next slide is, what do you think when you compare? So what do you get is that you get a difference that you do not know whether it's from the approach or from the perfusion, which activates a lot of things, and the sorting gates. So Nick's lab did the sorting gates, and we did the RiboTag IP, same mice, lysem-cre. The only difference was that they went for their traditional sort method. And so what you're going to look at is the enrichment over total liver RNA. And so in the black bars are the sort purified Kupffer cells. This is just their message. And then the RiboTag lysem-cre. And what you can appreciate is that almost everywhere, except for maybe these CCL, these chemokines, that there's a difference. And again, I can't tell you whether it's the population of cells you've sorted on or whether the act of perfusion has caused activation of these genes. We don't know. And I'm not saying that one approach is better than the other. I'm saying that it's important to have orthogonal approaches, whether it's immunohistochemistry, because this is a great approach to understand what's going on. But you do need complementary approaches because there's differences. So I haven't done the experiment that I really would have wanted, which is we treated with sorafenib, and I have the data on the next slide. So I apologize for that. Maybe next year, hey? Oops. So, yeah. So conclusions, and I'm going to go all the way back to the beginning, is that really the liver is the only mammalian organ that has the ability to generate. Oh, please tell me, what are the unique properties that allow hepatocytes and the non-parenchymal cells to do this? It's pretty amazing that it does it. There are redundant molecular pathways that enable the liver to regenerate. Again, I said middle of the talk that I have not yet seen a knockout, cell-specific or otherwise, that the animal doesn't regenerate. They may not report it in the paper, but the animals live. And so where is the necessary and sufficient pathway for liver regeneration? I think it has this amazing capacity to have redundancy, which, thank you, Pablo Neruda, lets us have this amazing organ that soldier on. As many of you know, is that the cells in the microenvironment are really important for normal liver regeneration and should be the focus of the antifibrotic, anti-angiogenic, and inflammatory strategies. Selective inhibition of signaling pathways in stroma may be best strategy. We talked about this. And then when flow is not an ideal approach, as it is in the animals that I was using, you need to have novel approaches. And again, I'm not saying the RiboTag or other tagged approaches are there, but I think that the field of liver regeneration could use this infusion of new approaches, right? How do we advance the field to ask old questions, right? So you use new technology to ask old questions, and this is a way to move the field forward. All right, so some thank yous. So there's a whole list at the University of Washington, and of course I have a photo montage on the next slide. UW, if anybody wants to work with Nick Crisp at the UW, he'd be happy to have you. A lot of other people that contributed to these projects that I didn't get a chance to talk about. I'm at the Fred Hutch Cancer Research Center now, and I work with Rob Pierce, and in data I couldn't show on our multispectral immunohistochemistry, Kimberly Smythe. The mice were originally made by Zymogenetics, which got bought by BMS. And then, of course, I've just got to harass Jordy for all his mentorship, and not so much, I think, is the phrase that he uses. So people that helped with these studies, and here in the center is Jordy when he came for a visit when Nelson was a bit sick. And here's Nelson in better days, where we were in my backyard enjoying a little popcorn. And then you can tell just by their posture who are the sort of lives of the lab. They're quite fun to work with, and so the rest. But before I leave, I just want to show you some of the work that we're doing in the immunopathology lab. And so this is this approach of using multispectral immunohistochemistry. So one of the things that we do at the Hutch is support clinical trials. We do it for the CAR T cell program. We're using NanoString to look at the tumor microenvironment to get a sense of what cells are there. But then we're doing this multiplex immunohistochemistry. And this was driven because the field knew with the CD8 T cells that it was where they were, not how many they were, right? Where they're positioned. And this came out of work that Rob did when he was at Merck. So it was like, where are they? They're on the outskirts of the tumor. So we know that the spatial network, the neighborhood where these tumor cells are, is important. And so in this, these two patients that have Merkel cell cancer, and so I thank Paul Neum and Kelly Paulson for these images, is that we stained them with cells that green pick up CD3, CD56, and in Merkel cell that picks up the tumor, which is this beautiful cyan. We've got CD8, HLA-DR, DAPI. You know, this is an ability to look, and you can look at the spatial proximity. Is the CD8 cell expressing PD-L1? And this is what we're doing with a lot of the CAR T cells, especially the ones that get added to the patient and transfused. Are they getting in? What do they look like? And so in order to get the handle on those cells, we need to generate new antibodies to them. And I've talked to a number of you about our love of llamas and the nanobodies, but this is what we're using them for, is to generate some new reagents. So I leave you with this because one of the lovely things about today was to talk about how we at the Fred Hutch could collaborate with both basic science as well as potential clinical trials in CAR T cells or other immunotherapy approaches. And I'm already going to have this great conversation because I think this is exactly what you need to do, Rafi, with your thymus, is that this is worth 1,000 words, right? So you can look, here's a thymus where you can look at this and you see this beautiful spatial network and learn so much about it. We can complement it with NanoString. We can complement it with laser capture microdissection. So I hope to take questions, but I want to leave you with the beautiful images of what we're doing now. So thank you very much, and bring the questions. What I can say is I don't know what is in response to what, but I know that we have a lot of inflammation. We have activation of stellate cells. We have a smoldering apoptosis, all of these which would bring in tissue and monocyte macrophages. And so we can quantify the difference between wild-type mice and the PDGFC transgenic mice, and it's a diverse population of cells. I know that the Kupffer cells increase in number if you believe that F480 is that specific, but we have an increase in a lot of other markers as well from array immunohistochemistry. So what causes what, I'm not going to commit myself. I think that there's definitely inflammation and an increase in these tissue resident macrophages as well as recruitment. Yes, I think that because we're in vivo and we treat, we don't know exactly whether the sorafenib, where the various targets are. It could hit any target that's available. We haven't done isolation of those cells and sort of treated. We have done a lot of work with cells in culture and adding sorafenib to them. And, monocyte macrophages seem to be the ones that get inhibited in kind of a dose response way. But this is cell lines versus inside. But yes, it's a formal possibility to say that change in the activation of the endothelial cells begets less inflammation and causes. I don't know how the epitope goes away, because it goes away at the gene level as well as the protein level. So we can go back to Glenn Merlino's model with the TGF alpha, and I think that in this case we've got a growth factor transforming growth factor alpha, and I think I agree with your hepatocytes proliferating and piling up on each other and just in an unhappy environment. There are a lot of other sort of mouse models of HCC that are a little bit better, but I think that I would say that there isn't a mouse model that I feel like is just the best recapitulation of what happens in the human, because what I would like to see is that there would be mutations that happen in those hepatocytes that come out of there. And we spent a long time trying to quantify mutations in tumors in the PDGFC mice, and this did not reveal a robust difference at the loci that we were looking at. So for me, it would be able to take that abnormal hepatocyte and actually take it from one animal and move it to another and have it, or into Marcus Grompi's FAH mice, and have it be an angry hepatocyte that just has nonstop proliferation. So I don't know of a model like that. Maybe the audience does, but I think these are just models that recapitulate some aspects of it. I think this is a very good philosophical question, is that if I thought that there would be a pathway that was the pathway, or maybe a couple pathways that would lead to the one that we need to manipulate to help diseased hepatocytes overcome their environment and proliferate. So we've been doing a number of knockout models to try and understand that, but they either compensate or they figure out another pathway that we can't quite see. So I think we move from, we can look at impaired and say that these are really important pathways, but this golden bullet, that this pathway, the EGF pathway, or any of the isoforms of these growth factors. So I guess I'm in a balance of that, is that I saw the SOS1, SOS2 stuff today, and I got pretty excited about that, because maybe this is now the sort of node. And so I was excited about the MIDI88 experiments because they're a node of signaling. And yet, the darn mice regenerated. Not my hands, your hands, Japanese group. I do know that the field spent a long time looking at IL-6 and IL-6 knockouts based on the Rebecca Taub report. Trautwein's group in Germany spent a lot. We've spent a lot of time on that, right? So it's like, do we go for a new pathway and say this is important? I think that almost any pathway we look at will have a role, right? There'll be a defect. How do you look at the constellation? You tackled this problem with trying to get multiple knockouts in the TLR, because we thought we would get them all by this, and sadly we didn't. So I don't know. I'm dodging your question, frankly. And this is my bias in drawing the cartoon, is that the center points were cytokines, their receptors, and the growth factors and receptors. But we know, like the glutathione reductase experiments, that we can change the timing, or Nelson force-fed amino acids, right? So we can push things through the TCA cycle in a different way. But we're talking about trying to take out glycolysis or gluconeogenesis, and this is going to be a challenge. So I think it's really important, but we just don't have the same number of knockouts on this. But I should say that I should correct the magnitude, because it is as equally important as the other pathways. You can't there's no more pinnacles. For a long time in the liver regeneration, we were so hepatocyte-centric, it was embarrassing to look back through the literature. And now we're not. And so we've evolved. We've evolved into this. So. So this is the real challenge with this project, is that we had so few mice because just being a SMAD3 knockout was detrimental to life. So we worked very hard. So I can't answer this very critical question that it said. Yeah. It really, the cell specific would be much better, much better. And they are there now. Yeah, I mean, the staining for the collagen is present. The genes are going to go down. So if you can metabolize the collagen, I think you have to have a second approach on this. But so the EMT, we didn't sort of go in with a specific set of markers, or the gene expression hasn't been analyzed for that. It's a good one. Thank you for the suggestion. Or the staining for this to look at this. This would complement this. And this is exactly a good thing because sorafenib does a lot of things.
Yeah, absolutely.
Yeah. So I think it's a dirty bomb. What appeals to me is the latter, that you have a nuanced sort of either dosing schedules so you may have sorafenib with another drug, but you may change the dosing schedule to try and change EMT or something like that. But you'd have something else that would be more specific, or you have a CAR T cell, right? And that you still have to deal with the fibrosis, right? That's not going to do that. That might target something. I think you're going to have a multiple approach. I'm hoping that Jordi and Barcelona will have a liver treatment staging diagram that we say, okay, here's what we have. But you did say inflammation in a lot of those cases, right? So we always this inflammation is kind of a constant. So I think that if we build the pyramid and we start with the base, then I would go for the inflammation and see how you can get that down. I think that would be a good one. Again, how the patient presents, when they present, how much reserve do they have? These are questions that are significant in how you approach. Jordi once said the best parameter was to say, hey, how are you feeling today? You know, we do our best. You do your best to find out what is the best way to go. And this interaction is a good one. Maybe the physical therapy, the get up and go test. But I think an inflammation in the final is a very good sort of foundational component, because I think you've got to get those non-parenchymal cells down in a lesser activated state. Those fibroblasts, the myofibroblasts, I mean, there's this beautiful paper from the Brenner group where they showed this epigenetic regulation, right? They could pull these cells out in a tickle of TGF-beta and they just went, right? So now we've got this problem of you've seen this disease and now you're fighting the battle on two fronts, new and the signal left from the last disease, the last state. So they're going to be prone and prior. So I'm going to pull out all arsenals. And I think that really we need to do parallel approaches and go after them. So that would be my.
Okay, well, thank you very much, Jim, for coming, and everybody for participating. And we look forward to the next liver seminars next month.
Yes. Thank you, everybody. Thank you very much. Thank you, Barcelona.